Hi, welcome to our channel of IGNOU audiobooks, Indira Gandhi National Open University, School of Continuing Education, SOCE, Master's Degree Programs, Master of Science, Food Nutrition, MSCDFSM, Revised, Second Year, MFN-004 Advanced Nutrition, Unit-4 Portions, 4.1 Introduction. The term protein, Greek, proteo to occupy first place, was first suggested by Berzelius to describe the complex organic nitrogenous substances found in animal and plant tissues. Proteins form three-fourths of the animal body on a moisture-free basis. They are essential for life processes. All the basic functions of life depend on proteins. Indeed, no form of life exists without proteins. They are found in every cell, make up the contractile elements and enzymes that catalyze the release of energy for the maintenance of life, and there is no physiological function in which they do not participate. Now, in this unit, let us get familiarized with the chemical nature, digestion, absorption, utilization, and other nutritional aspects of proteins. 4.2 Objectives. After studying this unit, you will be able to discuss the classification and functions of proteins, identify the food sources of proteins, explain the processes of digestion and absorption of proteins, recognize the methods of estimating the protein quality, as well as requirements at various stages of life, and describe the symptoms of protein deficiency. Proteins an overview. We just read that proteins are essential for maintaining and sustaining life. What are proteins, or what constitutes proteins that make it so important for us? Proteins contain carbon, hydrogen, nitrogen, sulfur, and some also contain phosphorus. In the Nutritional Biochemistry course, Unit 2, you may recall studying that proteins are chains of amino acids held together by peptide bonds. When a peptide chain is extended by more and more amino acids, until a chain length of 100 to several thousand amino acid residues is reached, it is classified as a protein. The nitrogen content of proteins varies from about 14 to 20%. The average value of 16% is used commonly for converting nitrogen content of foodstuffs or tissues into proteins by multiplying with the factor 6. 25, 100 by 16. I crude protein is equal to nitrogen 6.25. Let us now study and find out how proteins are classified. 4.2.1 Classification. Proteins vary widely in their properties. You may recall reading about the properties of proteins in the Nutritional Biochemistry course in Unit 2. We suggest you look up this unit once again now, as the information about proteins and their properties given there will supplement your understanding of the functions and other aspects of proteins discussed here in this unit. Let us consider the two very familiar proteins. One of them is the egg white protein, which is very sensitive. It denatures on heating, dissolves easily in water, and is quite reactive. While the other one is keratin of nails and hoofs, is wholly insoluble, tough and chemically inert, and resistant. Hence, it is not easy to classify proteins. We simply distinguish proteins which are insoluble and fibrous and function as structural material, scleroproteins, and globular proteins, those represented by egg white or serum proteins, which are soluble in water or salt solution and consist of spherical molecules. Besides classifying proteins on the basis of soluble and insoluble, proteins have been further classified based on the following attributes: classification based on chemical nature, classification based on chemical properties, classification based on amino acid content. This classification, let us get to learn about proteins based on this. A. Classification based on chemical nature. Frequently, proteins are classified based on the chemical nature of amino acids, such as solubility and prosthetic group, as simple, conjugated, and derived proteins. Let us get to know each of these. I. Simple proteins. Simple proteins are those which contain only amino acids or their derivatives and no prosthetic group. They yield only amino acids or their derivatives on hydrolysis. Let us see which are these and where are they found. 1. Albumins. proteins such as egg albumin and serum albumin are soluble in water and coagulable by heat. 2. Globulins. These proteins are insoluble in pure water, but soluble in neutral salt solutions. For example, serum globulin, tuberin, potato, arachin and conrakin, peanuts. 3. Glutenins. These are insoluble in all neutral solvents but soluble in very dilute acids and alkalis, e.g. glutenin of wheat. 4. Prolamins, proteins soluble in 70 to 80% alcohol, e.g. gliadin and zein. 5. Fibrous proteins. These proteins are characteristic of the skeletal structures of animals and also of the external protective tissues, such as the skin, hair, etc. Example, collagen, elastin and keratin. 6. Histones, soluble in water and insoluble in very dilute ammonia. On hydrolysis, they yield several amino acids, among which the basic ones predominate. The important proteins of this group are the thymus, histones and the globin of hemoglobin. Let us now move on to the next category of proteins, i.e. conjugated proteins and its types. 2. Conjugated proteins. Conjugated proteins contain some non-protein substances. Most proteins occur in cells in combination with prosthetic groups and hence are important for the nutritionist. These include 1. Glycoproteins. Most of the naturally occurring conjugated proteins are glycoproteins. Sugar molecules are covalently bound to them, especially those secreted from the cell. They range in size from a molecular weight of 15,000 to more than 1 million. The carbohydrate component varies from 1 to 85%. Glycoproteins with more than 80% of their molecules as carbohydrates are called proteoglycans. 2. Lipoproteins. These are the multi-component complexes of lipids and proteins that form distinct molecular aggregates. They contain polar and neutral lipids, cholesterol or cholesterol esters in addition to protein. The proteins and lipids are held together by non-covalent bonds. Lipids are primarily hydrophobic and cannot be easily transported through an aqueous environment as blood. The lipoprotein combination renders the lipid molecule hydrophilic and is transported in the blood to tissues which can use or store the lipids. 3. Nucleoproteins. Nucleoproteins are combinations of nucleic acids and simple proteins, which usually consists of a large number of basic amino acids. Nucleoproteins have very complex structures and numerous functional activities. All living cells contain nucleoproteins. Some cells, such as viruses, are composed of nucleoprotein. 4. Other conjugated proteins, the phosphoproteins and the metalloproteins are loose, as with phosphate-carrying protein, or tight, as with the phosphate in casein or the iron in ferritin, associations of proteins with phosphate groups or such ions as zinc, copper and iron. What are derived proteins? Let us find out next. 3. Derived proteins. Derived proteins are the derivatives of the protein molecule, apparently formed through hydrolytic changes in the molecule. These are either primary or secondary protein derivatives. Let us get to know them. 1. Primary protein derivatives. These are the derivatives of the protein molecule formed by hydrolysis involving slight alterations. Examples include A. Proteins. These are the insoluble products, which result from the incipient action of very dilute acids or enzymes, e.g. casein, curdled milk, fibrin, coagulated fibrinogen. B. Metaproteins. Proteins resulting from the action of acids and alkalis whereby the molecule is sufficiently altered to form proteins soluble in weak acids and alkalis, but insoluble in neutral solvents. C. Coagulated proteins. Insoluble proteins which result from the action of heat on protein solutions or the action of alcohol on the protein, e.g. cooked egg albumin or egg albumin precipitated by alcohol. 2. Secondary protein derivatives. These are the products of further hydrolytic cleavage of the protein molecule. Examples include A. Proteoses, soluble in water, not coagulable by heat, precipitated by saturating their solutions with ammonium. B. Peptones, soluble in water, not coagulable by heat, and not precipitated by saturating their solutions with ammonium sulfate. These represent a further stage of cleavage than the proteases. C. Peptides. These are the compounds containing 2 or more amino acids. An anhydride of 2 amino acids is called a dipeptide, one having 3 amino acids, a tripeptide, and containing several amino acids, a polypeptide. Peptides result from further hydrolytic cleavage of the peptones. Next, let us review the classification based on chemical properties. B. Classification based on chemical properties. Depending on their chemical properties and optical activity, the amino acids in proteins are classified under the following heads. 1. Monoamino monocarboxylic acids. Examples include glycine, alanine, valine, leucine, isoleucine, serine and threonine. 2. Monoamino dicarboxylic acids. Examples include aspartic acid and glutamic acid. 3. Diamino monocarboxylic acids, arginine and lysine. 4. Sulfur-containing amino acids, cysteine, cysteine, and methionine. 5. Aromatic and heterocyclic amino acids, phenylalanine, tyrosine, histidine, tryptophan, proline and hydroxyproline. The next classification is based on the amino acid content. Let us get to learn about this classification. C. Classification based on amino acid content. Nutritionally, amino acids are classified on the basis of the body's ability to synthesize them as essential, indispensable, and not synthesized in the body, and non-essential, dispensable, and that can be synthesized in the body, amino acids. Indispensable amino acids must be a part of the diet, while dispensable amino acids need not be present in food. These definitions, however, become blurred at the metabolic level. Lysine and threonine are perhaps the only metabolically indispensable amino acids because they are not transaminated to any nutritionally significant extent. This is a crucial point because lysine and threonine are the first and second limiting amino acids in cereal proteins. Lysine is the first limiting amino acid in human milk. Glutamic acid and probably serine are the only truly dispensable amino acids since these are the only amino acids which can be synthesized by the reductive amination of the appropriate keto acid. You may recall studying about these reactions in the nutritional biochemistry course in Unit 7. There is a third group of amino acids which are conditionally essential and are characterized by 2 features. Firstly, their synthesis uses other amino acids as carbon precursors and is confined to specific organs. This is an important metabolic distinction from the dispensable amino acids. For some conditionally essential amino acids, example, tyrosine, the precursor is a dispensable amino acid, while for others such as cysteine both an essential amino acid, methionine as sulfur donor, and a non-essential amino acid, serine are required. At the metabolic level, the organism's ability to synthesize a conditionally essential amino acid is constrained by the availability of a suitable amino acid precursor. Secondly, the maximum rate at which their synthesis proceeds may be limited and potentially restricted by developmental or pathophysiological factors. Thus, very low birth weight infants are unable to synthesize cysteine and proline and lack the ability to synthesize glycine. These factors are important because human milk proteins have low glycine content. Amino acids are classified based on the amino acid content as given in the Table 4.1. Table 4.1, Classification of amino acids. Look at the screen. We now know that proteins are composed of amino acids and that proteins differ in their amino acid makeup. Proteins lacking in one or more of the essential amino acids cannot be utilized to meet the protein requirements of the body, hence they are not good quality proteins. The nutritive value of a protein will be high if the amino acid makeup is very similar to that of the body proteins and will be low if it lacks partially or completely any one of the 10 essential amino acids, refer to Table 4.1, or if its amino acid composition is very much different from that of the body proteins. Based on their nutritive value or amino acids makeup, proteins are therefore classified as 1. Complete proteins. Example, egg proteins. These proteins promote growth and provide all the essential amino acids. 2. Partially complete proteins. Example, wheat proteins. These promote moderate growth and partially lack one or more essential amino acids, not prone. 3. Incomplete proteins. Example, gelatin or zein. They do not promote growth and completely lack one or more essential amino acids. After understanding how proteins are classified, let's move on to sources of protein in our next subsection. 4.2.2 Food sources. The important sources of proteins in the diets of low-income groups are cereals and legumes. Meat, fish, eggs and milk are important sources of proteins of high biological value. All seed meals and soy are rich potential sources of proteins. The protein content of some important foods is given in Table 4.2. Table 4.2, Protein content of some important foods, foods and protein, G100G, cereals, 313.27, cow's milk, 3.26, legumes, 1824, buffalo milk, 3.68, soybean, 37.8, tuna fish, 72.12, nuts and oilseeds, 2.7 to 23.6, goat liver, 20.32, egg, country hen, 72.96. We shall continue with our discussions on the metabolism and function of proteins. However, you must first attempt the check your progress exercise given below before proceeding further to recapitulate the learnt concepts. After having sound knowledge of what proteins are composed of, its types, and food sources, let us now study about their physiological aspect, i.e. digestion, absorption and transport in the body. 4.2.3 Digestion, absorption and transport. Under this subsection, we shall learn how proteins that we consume are digested, absorbed and transported to various body tissues. You may recall studying about this aspect in the Nutritional Biochemistry course in Unit 5, subsection 5.4.2. We suggest you look up this unit now. This will facilitate your understanding on this topic. A brief review on the process of digestion, the enzymes involved, their location and target amino acids is present next for your recapitulation. 4.2.3.1 Digestion. The daily protein intake of about 50 to 100 g, and the protein of enzymes, sloughed, bled or drop-off, epithelial cells, and mucins, which are found in the gut is almost completely digested and absorbed. This is a very efficient process and ensures a continuous supply of amino acids to the whole body's amino acid pool. The purpose of protein digestion is to liberate the amino acids of the consumed proteins. This is accomplished through a series of enzymes, which have specific target linkages as their point of action as shown in the Table 4.3. Table 4.3 Enzymes and their target linkages, enzyme their location and their target linkages, pepsin stomach peptide bonds involving the aromatic amino acids. Trypsin small intestine peptide bonds involving arginine and lysine, chymotrypsin small intestine peptide bonds involving tyrosine, tryptophan, phenylalanine, methionine and leucine, elastase peptidase A small intestine peptide bonds involving alanine, serine and glycine, carboxypeptidase A small intestine peptide bonds involving valine, peptidase A leucine, isoleucine and alanine, carboxy small intestine peptidase B peptide bonds involving lysine and peptidase B arginine. Endopeptidase aminopeptidase dipeptidase cells of brush D and tripeptides that enter the aminopeptidase brush border of the absorptive dipeptidase cells, the protein hydrolysis, called as peptidases, fall into two categories. Those that attack internal peptide bonds and liberate large peptide fragments for subsequent attack by other enzymes are called the endopeptidases, and those that attack the terminal peptide bonds and liberate single amino acids from the protein structure are called exopeptidases. The exopeptidases are further subdivided according to whether they attack at the carboxy end of the amino acid chain, carboxypeptidases, or the amino end of the chain, aminopeptidases. The initial attack on the intact protein is catalyzed by the intestinal epithelial cells. The hydrolysis of proteins in the gastrointestinal tract is completed by proteases secreted in the gastric juice and pancreatic juice, and also by proteases in the intestinal mucosa. In Table 4.3, you would have noticed that the digestion of protein starts only in the stomach. In contrast to carbohydrate and lipid digestion, which is initiated in the mouth with the salivary amylase and the lingual lipase, protein digestion does not begin until the protein reaches the stomach and the food is acidified with the gastric hydrochloric acid, HCl. Let us get to know about gastric digestion in greater details. Gastric digestion. If gastric HCl production is low and not adequate to maintain the pH of the stomach contents between 2 and 3, protein digestion in the stomach may be negligible. This will happen in aclohydria, a condition characterized by the failure of the intragastric pH to fall to less than 4. 0. Achelia gastrica, absence of gastric juice, partial or complete, or pernicious anemia. The HCl serves several functions in gastric digestion. It acidifies the ingested food, killing potential pathogenic organisms. However, not all pathogens are killed. Some are acid-resistant or are so plentiful in the food that the amount of gastric acidification is insufficient to kill all of the pathogens. HCl also serves to denature the food proteins, thus making them more vulnerable to attack by the proteolytic enzymes present in the gastric juice, namely pepsin and endopeptidase. It hydrolyzes peptide bonds in the interior of the protein molecule. Pepsin has a strong clotting action on milk. This factor is important in the digestion of milk proteins in infants. The optimum pH of pepsin action is about 2.0. Actually, pepsin is not a single enzyme. It consists of pepsin, which attacks peptide bonds involving phenylalanine or tyrosine, and several other enzymes which have specific attack points. The pepsins are released into the gastric cavity as pepsinogen. When the food entering the stomach stimulates HCl release and the pH of the gastric contents fall below 2, the pepsinogen loses a 44 amino acid sequence. The activation of the pepsins from pepsinogen occurs by one of the two processes, autoactivation and autocatalysis. Let's understand what these are. The first one is called autoactivation and occurs when the pH drops below 5. At low pH, the bond between the 44th and 45th amino acid residue falls apart, and the 44th amino acid residue, from the amino terminus, is liberated. The liberated residue acts as an inhibitor of pepsin by binding to the catalytic site until pH value of 2 is achieved. The inhibition is relieved when this fragment is degraded. Degradation occurs at pH 2 or by the action of pepsin. Thus, pepsin hydrolyzes mainly peptide bonds containing phenylalanine, tyrosine or tryptophan, and leucine and other acidic amino acids, and since food remains in the stomach for a limited time, dietary proteins are hydrolyzed mainly into a mixture of polypeptides as highlighted here with dietary proteins pepsin polypeptides. Let us get to know about the autocatalysis process next. Autocatalysis occurs when already active pepsin attacks the precursor pepsinogen. This is a self-repeating process and serves to ensure ongoing catalysis of the resident protein. The cleavage of the 44 amino acid residue, in addition to providing activated pepsin, also serves as a signal peptide for cholecystokinin release in the duodenum. This sets the stage for the subsequent pancreatic phase of protein digestion. Let us get to know about protein digestion in the intestine next. Proteolysis in the intestines. The main digestion of polypeptides produced in the stomach takes place in the intestines. Cholecystokinin stimulates both the exocrine pancreas and the intestinal mucosal epithelial cells to release its digestive enzymes. The proteases involved in the digestion are trypsin, chymotrypsin, and carboxypeptidase, as highlighted in the table 4.3, secreted in pancreatic juice, and aminopeptidases present in the intestinal mucosa. The intestinal cells release an enzyme, enteropeptidase or enterokinase, which serves to activate the protease trypsin, released as trypsinogen by the exocrine pancreas. This trypsin not only acts on food proteins, it also acts on other proteases released by the exocrine pancreas, thus activating them. Thus trypsin as an endoprotease chymotrypsinogen by releasing chymotrypsin, on proelastase by releasing elastase, and on procarboxypeptidase by releasing carboxypeptidase. Trypsin, chymotrypsin, and elastase are all endoproteases, each having specificity for particular peptide bonds. Trypsin and chymotrypsin act at pH 7.4 to 8.0. Trypsin hydrolyzes mainly peptide linkages containing tyrosine or phenylalanine proteins and polypeptides. Trypsin and chymotrypsin peptides amino acids. Each of the three proteases, i.e. trypsin, chymotrypsin, and elastase have serine as a part of their catalytic site, so that any compound that ties up the serine will inhibit the activity of these proteases. One such inhibitor, disopropylphosphofluoridate, reacts with serine and stops protein digestion. Through the action of pepsin, trypsin, chymotrypsin, and elastase, numerous oligopeptides are produced, which are then attacked by the amino and carboxypeptidases of the pancreatic juice and those on the brush border of the absorptive cells. Carboxypeptidase A hydrolyzes the end group in peptides containing aromatic or aliphatic amino acid, thus releasing free amino acids as shown herewith. Carboxypeptidase B hydrolyzes peptides containing arginine and lysine residues. The intestinal mucosa contains a group of aminopeptides which complete the hydrolysis of peptides to amino acids. The intestinal mucosa also contains tripeptidase, dipeptidase, etc., which hydrolyze tri- and dipeptides as highlighted in the reaction given herewith. 4.2.3.3 Transport of amino acids. More than one transport or carrier system functions in the absorption of amino acids. The active carrier system for neutral amino acids shares a common membrane carrier. Neutral amino acids and those with the short or polar side chains, serine, threonine, and alanine, are transported by the shared carrier system. The basic amino acids, that is, lysine, arginine, and histidine, share a carrier system with cysteine. The mechanism of transport and uptake are depicted in Figure 4.1. Figure 4.1, carrier-mediated sodium-dependent amino acid transport. In Figure 4.1, you may have noticed that the amino acid transport is dependent on the Na ion. The dependence of amino acid transport on Na ion suggests a direct interaction between the carrier and Na ion. This is similar to that observed in the absorption of glucose, as you may recall studying in the last unit. The amino acid associates with the carrier and Na ion in the microvilli, and the complex travels to the inner side of the membrane where it dissociates, releasing the amino acid and Na ion into the cytoplasm as illustrated in Figure 4.1. Thus the amino acid leaves the absorptive cell with sodium. The carrier, Na ion, is recirculated back to the lumen for reuse. As sodium enters the cell, potassium is pumped out via a Na-K-ATPase system. As sodium leaves the cell, potassium flows back in and the electrolyte balance is maintained. The Nat ion is then actively transported out of the cell, so we have looked at the digestion, absorption, and transport of proteins and amino acids. What are the functions of proteins in our body? Let us study and find out. Next, 4.2.4 Functions. Each of the various proteins in the body serves a specific function in the maintenance of life. Any loss in body protein, in reality, means a loss in cellular function. In contrast to lipids and carbohydrates, proteins have no true body reserve. Humans, when deprived of or insufficiently supplied with protein, compensate for this dietary deficiency by catabolizing some, but not all, of their tissue functionality. Cells, tissues, organs, and whole systems cannot exist without proteins serving their various functions. So let us get to know the varied functions of proteins in our body. We begin our discussion on functions, first with the bodybuilding function. 1. Bodybuilding functions of proteins. The primary functions of proteins, as you might be aware, is tissue growth and maintenance. Protein contains amino acids, the building blocks that our bodies use to build and maintain muscles, bone, skin, blood, and other organs. Thus, proteins play an important role in growth and bodybuilding. For the constant growth of human beings from birth to adulthood, a regular supply of dietary protein is required. Now, this does not mean that the body does not require proteins once the growth ceases. During adulthood, worn-out cells, body tissues need continuous replacement. Proteins are required for maintenance of our body. Proteins, therefore, are crucial and required throughout our lifespan for growth. Bodybuilding and maintenance. 2. Protein as an energy source. Proteins contribute to the body's energy need. If diet does not furnish enough calories from carbohydrates and fats, proteins are catabolized to give energy. 1 gram protein yields 4 kcal. But what is important for us is to understand is that this is not the major function of proteins. This only happens when, as mentioned earlier, the diet does not supply enough energy-giving nutrients. 3. Proteins as enzymes. From conception to death, living cells use oxygen and metabolize fuel. Cells synthesize new products, degrade others, and generally are in a state of metabolic flux. For these processes to occur, catalysts are needed to enhance each of the many thousands of reactions occurring in the cell. These catalysts, called enzymes, are proteins, as you have already studied in the nutritional biochemistry course in Unit 4. Enzymes make up the largest and the most specialized class of proteins. Each enzyme is unique and catalyzes a specific kind of reaction. In the cell, enzymes are found in cellular compartments, cytoplasm, nucleus, mitochondria, etc., as well as the membranes within and around the cell wall. The location of an enzyme is one of its characteristics and dictates, in part, its role in metabolism. Many enzymes are complex proteins. They consist of a protein component and a prosthetic group. The protein part is called a enzyme, and the prosthetic group, coenzyme, as illustrated in Figure 4. 2. A detailed discussion on enzymes is presented in the Nutritional Biochemistry course, Unit 4. We suggest you read this unit now for a better understanding of the functions of proteins as enzymes. Figure 4.2. Holoenzyme. Enzymes consist of specific sequences of amino acids. The catalytic function of an enzyme is intimately related to its amino acid sequence. Enzymes must possess a shape that will complement the reactive molecular shape of the substrate in the same way as a key fits into a lock. This is commonly referred to as lock and key mechanism. This shape is a function of the enzyme protein's primary, secondary, tertiary, and quaternary structure. In the same way, substrates should also have specific shapes in order to be catalyzed by their respective enzymes. This is the reason why only D sugars or L-amino acids can be metabolized by mammalian cells. These stereoisomers conform to the shape required by the enzyme, which serves as its catalyst. While enzymes show absolute specificity, the specificity generally applies to the entire molecule. If, however, the substrate is large and complex, the structural requirements are less stringent in that only that part of the substrate involved in the enzyme-substrate complex should have the appropriate molecular arrangement. The portion of the substrate not involved in the reaction need not be the appropriate conformation. Some enzymes are specific for only one substrate, others may catalyze several related reactions. While some are specific for a particular substrate, others are specific for certain bonds. This is called group specificity. For example, glycosidases act on glycosides, pepsin and trypsin act on peptide bonds, and esterases act on ester linkages. Within this group, certain enzymes exhibit greater specificity. Chymotrypsin preferentially acts on peptide bonds, in which the carboxyl group is a part of the aromatic amino acids, phenylalanine, tyrosine, or tryptophan. Enzymes such as carboxypeptidase catalyze the hydrolysis of the carboxy terminal or amino-terminal amino acid of a polypeptide chain. This bond specificity, rather than molecular specificity, is useful to the animal in that it reduces the number of enzymes needed within the organism. Incidentally, the above enzymes are very useful to the protein chemist in his/her determination of the amino acid sequence of a given protein. Cells synthesize enzymes in much the same fashion as they synthesize other proteins, yet enzymes are relatively short-lived. Cells must continually synthesize their enzymes if they are to survive. So, we have looked at the functions of proteins as enzymes. Proteins also function as carriers in the body. Let us get to know about this function next. 4. Proteins as carriers. A large variety of compounds are carried in the blood between tissues and organs of the body. Some of the compounds require specific protein for their transport. Not only is this specific protein necessary for the transport of compounds insoluble in blood, but it is also necessary to protect these compounds from further reactions that take place during the transport process. Some of the membrane proteins are carriers, and some are both carriers and enzymes. Both intracellular and extracellular carriers have been identified. The plasma proteins which can have a carrier function are the albumin and the A and B globulins. The best studied of the plasma carriers are those associated with the transport of lipid, called lipoproteins, since these lipoproteins, carriers plus lipids, when levels are elevated, appear to be related to the development of a variety of diseases. These lipoproteins comprise of about 3% of the plasma proteins. They are the loose associations of such lipids as phospholipids, triacylglycerols, and cholesterols, and represent an example of how proteins function as carriers. The lipids they carry are either from the diet or are synthesized de novo in tissues, such as the liver. The B-globulin proteins carry these lipids to such sites as muscle or adipose tissue, where they are either used or stored. The release of the lipid from the protein carrier is a complicated process. In adipose tissue, the lipoprotein is attached to a membrane receptor site and enzyme. Lipoprotein lipase cleaves the lipid from the protein. The lipid is then picked up by another protein called a lipid-binding protein and is carried to the interior of the cell for storage. The globulin protein carrier, once free of its lipid, returns to the liver or intestinal mucosa and is recycled. The plasma lipids, phospholipids, acylglycerols, cholesterol, cholesterol esters, and free fatty acids are usually transported as loosely associated lipid-protein complexes. At least 3 different proteins have been identified. Albumin usually transports the free fatty acids, whereas the A and B globulins transport the phospholipids, acylglycerols, and cholesterols. The different lipoprotein complexes can be separated and identified on the basis of their antigenicity, their electrophoretic mobility, and their density. The low-density or B lipoproteins contain the B peptide, cholesterol, and some phospholipids. The majority of phospholipids are carried as lipoproteins. With age, the lipid content of the plasma tends to rise, and the rise is reflected almost entirely as an increase in B lipoproteins. As the density of the lipoproteins decreases, the molecular weight and complexity of the lipid it carries decreases. The A lipoproteins carry mainly, up to 60%, acylglycerols. These glycerols are usually those synthesized in the body rather than coming from the diet. The dietary acylglycerols are usually carried as chylomicrons. These particles are the largest and least dense of the lipid-protein complexes. Read Unit 7 in the Nutritional Biochemistry course for more information, in addition to serving as carriers of lipids. Some of the globulins in the plasma can combine with iron and copper, as well as with other divalent cations. These combinations are called metalloproteins. The globulins serve to transport these cations from the gut into the tissues where they are used. The monovalent cations, sodium and potassium, do not need carriers, but most other minerals do. Many hormones and vitamins require transport or carrier proteins to take them from their point of origin to their active site. In addition, there are intracellular transport proteins such as the lipid-binding proteins that are responsible for the transport of materials between the various cellular compartments. Lastly, there are transport proteins which carry single molecules. The classic example is hemoglobin, the red cell protein, responsible for the transport of oxygen from the lungs to every oxygen-using cell in the body. From carrier function, we move on to the regulatory function of proteins. 5. Proteins as regulators of water balance. As substrates and solutes are transferred or exchanged across membranes, water has a tendency to follow to maintain equal osmotic pressure on each side of the membrane. If osmotic pressure is not maintained, the individual cells either shrink from lack of internal water or burst from too much. You may recall studying about this phenomenon in the Food Microbiology and Safety course in Unit 3. The balance of water between intracellular and extracellular compartments is closely regulated. Water balance across the capillary membrane is carefully controlled. A close balance is maintained between the osmotic pressure of the plasma, the interstitial fluids, and the cells, and the hydrostatic pressure exerted by the pumping action of the heart. The total osmotic pressure of the plasma and of the intra- and extracellular fluids is the result of its content of inorganic electrolytes, its organic solutes, and its proteins. The concentrations of the electrolytes and organic solutes in plasma, interstitial fluid and cells are substantially the same, so that the contribution to the osmotic pressure by these substances is practically equal. However, since there are more proteins in plasma than in the cells, plasma exerts an osmotic pressure on the tissue fluids. The result of this inequity of solutes is the drawing of fluids from the tissue spaces and from the cells into the blood. Opposing this force is the hydrostatic pressure, exerted by the pumping action of the heart, which moves fluids from the blood into the tissue spaces and into the cells. The hydrostatic pressure is greater on the arterial side of the capillary loop than on the venous side. There is some kind of interplay between these four kinds of pressure: blood osmotic pressure, tissue osmotic pressure, blood hydrostatic pressure, and tissue hydrostatic pressure. This interplay results in the filtration of solutes and metabolites and the transfer of oxygen from the arterial blood into the tissues and cells it supplies, and on the venous side, a resorption from the tissue space of CO2, metabolites, and solutes back into the blood supply. Albumin plays a more significant role in maintaining the osmotic pressure than the other blood proteins because of its size and abundance. It is a small molecule and has a greater number of particles per unit volume than the other larger serum proteins. With fewer proteins in the serum, water leaks out into the interstitial space and accumulates. You may be familiar with this condition, commonly referred to as edema. The edema of protein deficiency may also be the result of the body's inability to regulate the protein hormone, particularly antidiuretic hormone, ADH. This hormone plays a role in controlling water balance. The effect of protein is on the distribution of water amongst the various body compartments than on the total body water. So now you have understood what important role proteins can play in regulating the water balance. Proteins also function as biological buffers. What do we mean by this, and how proteins function as biological buffers is discussed next. So now you have understood what important role proteins can play in regulating the water balance. Proteins also function as biological buffers. What do we mean by this, and how proteins function as biological buffers is discussed next. 6. Proteins as biological buffers. Proteins have the ability to accept or donate hydrogen ions, and by doing so they serve as biological buffers. You may recall studying under the properties of proteins in subsection 2. 6.2 in Unit 2 in the Nutritional Biochemistry course. In blood, there are three important buffering systems: plasma proteins, hemoglobin, and carbonic acid bicarbonate. The equilibrium reactions for each of these buffering systems is as follows. H protein, H.co, HHCO, HHB. The first of these buffering systems, the plasma proteins, functions as a weak acid-salt buffer when the free carboxyl groups on the protein dissociate, or as a weak base-salt buffer when the free amino groups dissociate. Although the buffering ability of the plasma protein is extremely important in maintaining blood pH, it is not as important as the other two systems. The second buffering system, carbonic acid bicarbonate, is extremely effective because there are reactions which follow this equilibrium which will regulate either acids or bases. The H.co level in plasma never goes too high because it is in equilibrium with CO2, Co to H2O, which is expired by the lungs. In blood, this equilibrium proceeds very quickly because of the presence of carbonic anhydrase, an enzyme found in red blood cells which catalyze it. If the carbonic acid bicarbonate reaction goes in the opposite direction, the concentration of the HCO so formed will be regulated by the kidneys. The third important buffering system in blood results from hemoglobin. Hemoglobin has 6 times the buffering power of the plasma proteins. It functions well as a buffer because it is present in large amounts. It contains 38 histidine residues. Histidine residues are good buffers because they can dissociate to H and the imidazole group, and because hemoglobin exists in blood in two forms, reduced hemoglobin and oxyhemoglobin. It is thus a weaker acid and a better buffer. Next, let us look at the function of proteins as structural elements. 7. Proteins as structural elements and structural units. The liver cell membrane analysis shows that this membrane contains 50 to 60% protein, 35% lipids, and 5% carbohydrates. The carbohydrate present is joined primarily to the protein-forming glycoproteins, compounds which constitute the receptor sites of several hormones. The protein portion of the membrane is so oriented that its hydrophilic aspects are also in close proximity to the intracellular and extracellular fluids. The protein molecules are interspersed within the lipids and lend both structural stability and fluidity to the membrane. Membrane function depends on how the proteins are placed in the membrane and on the fluidity, which results from the combination of proteins in a lipid mixture. If the lipid is more saturated, a more rigid crystalline structure will form. Many lipids, being fluid and less rigid, allow the proteins to change their shape in response to ionic changes, and hence these proteins function as enzymes, carriers, binding or receptor sites, or entry ports for a large variety of materials binding, entering or leaving the cell. Thus, proteins serve as the structural and functional units of the cell membrane. Proteins are also important intracellular structural units. Muscle is composed of 20% protein, 75% water, and 5% inorganic material, glycogen, and other organic compounds. The major proteins in muscle are myosin, a large globular protein, and actin, a smaller globular protein. These two proteins, plus the filamentous tropomyosin and troponin, are the molecular components of the muscles. The muscle proteins are characterized by their elasticity, which contributes to the contractile power of the tissue. The most important structural function of protein is related to skin and connective tissue. The skin is composed of epithelial tissue which covers not only the exterior of the body but also lines the gastrointestinal tract, respiratory tract, and the urinary tract. One of the major proteins found in the skin is melanin. Melanin is a tyrosine derivative and provides the pigmentation or characteristic color to the skin. Persons unable to form this pigment are albinos, and the disease is called albinism. Keratin is the protein which forms hair, nails, hooves, feathers, or horns. Each of these structures is slightly different but all contain keratin. This protein is insoluble in water and is resistant to most digestive enzymes. It has a high percentage of cystine. Connective tissue is that tissue which holds various cells and tissues together. It includes bones and teeth also, since they start with a matrix protein into which various amounts of minerals are deposited. Collagen and elastin are the two distinct types of proteins in the connective tissue. It contains proline, hydroxyproline, and glycine. These proteins are not easily degradable and are inert metabolically. Even in protein-deficient states, the body will synthesize collagen and elastin, and these proteins will not be catabolized for needed amino acids. However, this protein can be degraded to a limited degree by boiling in acid. It is then converted to gelatin. The collagen of bone, skin, cartilage, and ligaments differ in chemical composition from that of the white fibrous tissue which holds individual cells together within muscle, liver, and other organs. Elastin and chondroalbumoid are two other proteins in the connective tissue. They are present in small amounts and serve as a part of the structural protein. Proteins as structural elements have been studied above. Interestingly, proteins also surround the joints and function as lubricants. Let's look at this lubricant function of proteins next. 8. Proteins as lubricants. The mucus of the respiratory tract, oral cavity, vaginal tract, and the rectal cavity reduces the irritation which might be caused by materials moving through these passages. This mucus is a mucoprotein, a conjugated protein which contains hexosamine. Proteins as lubricants also surround the joints and facilitate their movement. The absence of these lubricants or substantial decrease in their fluidity through deposition of minerals makes skeletal movement difficult and painful. Last but not the least, proteins also have an important role to play in the immune system. Let us find out how. 9. Proteins in the immune system. Proteins such as Y globulins serve to protect the body against foreign cells. The immunoglobulins produced by lymphocytes are the large polypeptides having more than one basic monomeric unit. These proteins differ in their amino acid structure, which in turn affect their secondary, tertiary, and quaternary structures. Just as the amino acid sequence of an enzyme determines substrate specificity, the amino acid sequence of the immunoprotein assures antigen-antibody specificity. The initiation of synthesis of particular immunoglobulins by the lymphocytes requires the binding of an antigen, a foreign protein, to the cell surface at particular locations called antigen receptors. The immunoglobulin synthesized binds with the foreign protein immobilizing it. Thus, the complex antigen-antibody is formed about which we have already studied in great details in the Applied Physiology course in Unit 3. Do look up this unit now to understand this process. With this, we come to an end of our study on the functions of proteins. With a clear understanding on the functions of proteins, next we shall review the methods of determination of proteins and amino acid content in foods. 4.3 Methods of determination of proteins and amino acid content in foods. The methods for protein quality evaluation are grouped under the following headings. I. Analytical methods for the determination of nitrogen and amino acids in foods. 2. Chemical and microbiological assays of protein quality. 3. Evaluation of protein quality in experimental animals. 4. Clinical methods for evaluation of protein quality, OPLEs. In this section we shall review both the qualitative and quantitative evaluation of protein and amino acid content in foods. We shall begin with the quantitative analytical method for the determination of nitrogen and amino acid in foods. Quantitative analytical method for estimation of nitrogen and protein. You may recall reading earlier that proteins contain nitrogen along with carbon, hydrogen, and some other substances. Nitrogen, N, in food not only comes from amino acids in protein but also exists in additional forms that may or may not be used as a part of the total nitrogen economy of humans and animals. Thus, determination of nitrogen is commonly used to determine the protein content of a sample. Total nitrogen, which is determined by the Kellahal procedure, is converted to crude protein by a factor of 6.25, as mentioned earlier. Thus, the conventional measure of protein or crude protein in foods is an X 6.25, and it is recommended that this one factor be used in nutritional studies in which whole diets contain more than one source of nitrogen, the Keldahl's method is commonly used in estimation of protein content of foods and has been extensively used for protein estimation of various foodstuffs. Although over a period of time many other methods have emerged for determination of organic nitrogen, this method still remains an old favorite because it is reliable and has very well standardized procedures. We shall not go into the details related to the method of this process here in this unit, since it has already been covered in the Nutritional Biochemistry Practical Course, MFN-002, in Practical 5. We suggest you look up this practical now. Next, we shall review the methods for protein quality evaluation, qualitative estimation of protein amino acid content in foods. The protein and amino acid content of foods helps to interpret nutritional differences among foods in terms of their amino acid makeup. This has been obtained only through experiments on animals or human beings. The methods available to determine protein quality are based on several parameters. They are categorized as methods based on I, growth and body weight changes. The methods under this category include times protein efficiency ratio, per, net protein ratio, NPR, gross protein value, times rat repletion method, nitrogen growth index, slope ratio method, 2, carcass nitrogen analysis. This method includes nitrogen retention method, net protein utilization, NPU, times rat repletion methods, 3. Nitrogen balance includes nitrogen balance, digestibility coefficient, biological value and net utilization of dietary proteins, nitrogen balance index, egg replacement method. 4. Regeneration of blood and liver constituents includes liver protein regeneration, blood protein regeneration, regeneration of liver enzymes. Availability of amino acids includes chemical methods, times enzymic methods, microbiological methods, animal assays. 6. Miscellaneous parameters include plasma amino acid levels, microbiological methods. 7. Chemical scoring include chemical score, essential amino acid index, times simplified chemical score. Qualitative differences in food protein sources can be determined by many methods. The traditional methods protein efficiency ratio, per, net protein utilization, NPU, and biological value, BV, per, NPR and NPU have been described in brief, followed by discussion on the more recent methods of estimation using the fecal protein and ileal amino acid, IA, digestibility to compute the protein digestibility corrected amino acid score, PDCAS, and digestible indispensable amino score, DIAS. Let us get to know these methods. We shall start off with per I.E protein efficiency ratio. Protein efficiency ratio, per method was developed by Osborne, Mendel and Ferry in 1919 and is based on the growth of young rats. The diets usually contain 10% of dietary protein to be tested and are complete in all other dietary essentials. Groups of albino rats, 21 days old, are fed for a period of 4 weeks on different diets. Records of the gain in body weight and protein intake of the rats are maintained. The protein efficiency ratio, per, is calculated using the following formula. PER is equal to gain in body weight, G, upon protein intake, G, is equal to gain in weight per G of protein consumed. Per, therefore, is a measurement of the efficient utilization of the proteins in the body. You would be interested to know that the egg protein has the highest protein efficiency ratio. Hence, egg protein is the standard to which all other forms of proteins are measured. Next, let us learn about digestibility coefficient. Digestibility coefficient. You have earlier learned that dietary proteins are hydrolyzed to amino acids during digestion. The digestion begins in the stomach by the action of pepsin and later completed in the intestines by trypsin, erepsin and other enzymes. Proteins differ in their digestibility. Egg and milk proteins are easily digested and converted into amino acids while pulse proteins are slowly digested to amino acids. Amino acids are absorbed into the bloodstream and the unhydrolyzed portion of the protein is wasted in the faxes. Thus, digestibility coefficient of protein refers to the percentage of the ingested protein absorbed into the bloodstream after the process of digestion is after the process of digs complete, when an animal is fed on nitrogen. N. Free diet, certain amount of nitrogen is excreted in the faeces. This is derived mainly from the digestive juices. This is called endogenous fecal N. When a protein food is given, the N found in feces consists both of endogenous N and food N lost in digestion. To find out N lost in digestion, endogenous fecal N should also be determined. For the determination of the digestibility coefficient, therefore, the data on food nitrogen intake, IN, total fecal nitrogen excreted, FN, and endogenous fecal nitrogen, FE, are required. The digestibility coefficient can thus be calculated using the formula digestibility coefficient is equal to 100 into N intake minus N in feces minus endogenous fecal N upon N intake is equal to 100 into IN into Fn minus Fe upon In, where FnFe is the food nitrogen lost in digestion. N intake. The digestibility coefficients of proteins are influenced by several factors, such as the presence of indigestible carbohydrates like cellulose and hemicellulose and the presence of proteolytic enzyme inhibitors. A term which is closely and synonymously used with protein quality is biological value of protein. Biological value. A method for determining the biological value of proteins was developed by Mitchell in 1925. It measures the quantity of dietary proteins utilized by the animal for meeting its protein needs for maintenance and growth. Groups of albino rats, 28 days old, are fed successively on the following diets for a period of 10 days: 1. Protein-free diet, and 2. Diet containing 10% protein to be tested. Urine and feces are collected by keeping them in metabolism cages. Records of food intake are maintained. The diet, urine and feces are analyzed for nitrogen. Biological value is then calculated using the following formula. Look at the screen, times 100. Thus, BV is a measurement of protein quality expressing the rate of efficiency with which the protein is used for growth. On a scale with 100 representing top efficiency, Table 4.4 presents the biological values of proteins in several foods. Table 4.4, biological values of proteins in several foods: whole egg 93.7, milk 84.5, fish 76, beef 74.3, soybean 72.8, rice, polished 72.8, wheat, whole 64, corn 60, beans, dry 54. Amino acid score. A is the amino acid score determines the effectiveness with which absorbed dietary nitrogen can meet the indispensable amino acid requirement at the safe level of protein intake. By this approach, we can evaluate the capacity of a given protein or mixture of proteins to meet the essential amino acid and nitrogen requirement of an individual. Amino acid score is expressed as the ratio of mg of amino acid in 1g of test protein to mg of amino acid in reference protein. Amino acid score, AS, is equal to mg of amino acid in 1g of test protein upon mg of amino acid in 1g reference protein. The 2007 FAO-WHO UNU consultation has recommended the standard amino acid pattern presented in Table 4.5. Retained by ICMR 2020, for scoring quality of protein. Using this reference requirement pattern, the amino acid score for a test protein can be calculated. Table 4.5, amino acid scoring pattern for toddlers, children, adolescent, and adults, mg protein, amended values from the 2007 WHO FAO-UNU report, values based on the amino acid requirement and the protein requirement for each age group. Histidine, ILE, isoleucine, leucine, LYS, lysine, SA, sulfur amino acids, AA, aromatic amino acids, DARE, threonine, TRP, tryptophan, WAL, valine source, dietary protein quality evaluation in human nutrition. Report of an FAO expert consultation FAO 2013, With our understanding of the BV and amino acid score, we move to the concept of protein digestibility corrected amino acid score, PDCAS. PDCAS, it is a chemical score which is derived from the ratio between the first limiting amino acid in a test protein and the corresponding amino acid in a reference amino acid pattern and corrected for true fecal indigestibility. It is expressed as, then, protein digestibility corrected amino acid score, PDCAS, is equal to protein digestibility into amino acid score, PDCAS, percent, is equal to true protein digestibility, percent, is equal to mg of amino acid in 1g of test protein into 100 upon mg of amino acid in 1g reference protein. PDCAS value for some protein sources is given in Table 4.6. Table 4.6, PDCAS of some protein sources, digestible indispensable amino acid score, DIAS. Digestible indispensable amino acid score, DIAS, is a protein quality index which is based on true ileal, terminal end of small intestine, amino acid digestibility. It provides for a more accurate measure of the amount of amino acid absorbed by the body and the protein's contribution to human amino acid and nitrogen requirement. DIAS has replaced the current protein ranking standard. The protein digestibility corrected amino acid score, PDCAS. DIAS is defined as follows: DIAS percent is equal to 100 mg of digestible dietary indispensable amino acid in 1g of the dietary protein, mg of the same dietary indispensable amino acid in 1g of the reference protein, recommended amino acid scoring patterns, I amino acid pattern of the reference protein, to be used for calculating protein quality for dietary assessment for different age groups is presented in Table 4.5. In calculating DIAS, the ratio should be calculated for each dietary indispensable amino acid and the lowest value designed as the DIAS. Net protein utilization, NPU, Mitchell, 1922, introduced the term net utilization of dietary protein, which is a product of digestibility coefficient and biological value divided by 100. As shown herewith, NPU is equal to digestibility coefficient into biological value upon 100. A direct method of estimating net protein utilization, NPU, was developed by Miller and Bender, 1955. Groups of albino rats 28 days old were used. One group was fed on a non-protein diet while the other groups were fed on the test diets containing different proteins at 10% level for a period of 10 days. The food intakes of the animals were measured. The animals were killed at the end of 10 days and the body nitrogen was determined by Kjeldahl method on a sample of dried and powdered carcass. The NPU is calculated according to the formula. NPU is equal to body N of the test group minus body N of the non-protein group N consumed by non-protein group UPO N consumed by test group into 100. The main advantage of determining NPU of a food or diet is that it helps in the calculation of the net available protein of the diet. Studies have shown that there is a good correlation between the values for per NPU and chemical score. Let us now understand what is a net protein ratio or NPR and how is it determined. Net protein ratio, NPR. This method was introduced by Bender and Doyle, 1957, and is a modification of the per method. In this method, an allowance is made for the protein requirements for maintenance. The method consists of feeding a group of weanling rats on a diet containing 10% of the test protein and another comparable control group on a non-protein diet for a period of 10 days. The NPR is calculated by adding the loss in weight of the control group to the gain in weight of the test group and dividing the total weight, G, by the quantity of protein consumed by the test group according to the following formula. NPR is equal to gain in weight, G, of the test group loss in weight, G, of the non-protein group upon protein intake, G. NPR values have been reported to correlate closely with NPU values. Lastly, nutrient-to-energy ratio has attracted wide interest as indices of dietary quality. The protein-energy ratio as a measure of dietary quality is explained next. protein-energy ratio, NDP call percent, Platt and his colleagues, 1961, are largely responsible for the introduction of the ratio of protein energy to total energy, pay ratio, as a useful measure of dietary quality in human nutrition. To take into account both the quality and concentration of the protein, they introduced the concept of the net dietary protein calories as a percentage of total calories, NDP call percent. The NDP call percent is the net dietary protein value expressed as percent of total calories. The NPU or the chemical score could be utilized in calculating the NDP call percent, as both of them are indicative of protein value. The formula used is as under. NDP call percent is equal to protein calories in the diet upon total calories into NPU into 100, or NDP call percent is equal to protein calories in the diet upon total calories into amino acid score into 100. For adults, diet with an NDP call percent of 5% would be adequate to maintain health. In infants, children, adolescent, and pregnant women, growth is supported only by a diet providing an NDP call percent of 8% or above. Now that we have studied and understood the various methods of determining proteins, it is important that we practically assess the protein amino acid quality of our diet or perhaps of other population groups. To help you in this task, we have included various activities in the practical manual, MFN-004, accompanying this course. So go ahead, carry out these activities. Let us now proceed further and find out the ways by which we can improve protein quality of our diets. 4.4 Improvement of quality of protein in the diet. Since the net protein utilization, NPU, values of milk or egg proteins are higher than those of proteins of average diets consumed in different countries, a correction has to be made for this variation in the NPU of dietary proteins. This is shown here with dietary protein requirements, G, is equal to safe level of intake of egg or milk proteins into NPU of egg or milk proteins upon NPU of dietary proteins. For example, if the NPU of the dietary proteins is 45 and egg protein is 90, the correction factor will be 2. In other words, the amount of this dietary protein needed to satisfy the requirements of a given population group will be twice as high as that of egg or milk protein. The next thing that might come to your mind is regarding the protein quality of the Indian diets. Average Indian diets, as consumed in different parts of the country, consists chiefly of vegetable source of protein. The amount of animal protein depends on the diet habits, with only milk providing a source of animal protein in vegetarian diets to varying amounts of meat and flesh in the non-vegetarian diets. The question of whether the protein quality of our predominantly vegetarian diets is adequate and what are the ways in which the quality of protein in our diets can be improved has been addressed through a number of studies. Habitually, Indian diets are cereal-based diets, limiting in lysine, an essential amino acid critical for growth and development in children. The term limiting is used to describe that indispensable amino acid which is present in the lowest quantity in the food, in comparison with the same amino acid in a reference protein such as egg or milk. The quantity of the amino acid expressed in terms of per g nitrogen. Further, the diet is predominantly vegetable-based, and foods of animal origin do not usually find a place because of their high cost. A large percentage of people are vegetarian and their diets include pulses, vegetables, cereals, and grain products. These plant foods tend to have too little of one or more indispensable amino acids, i.e. lysine, threonine, tryptophan, or methionine, particularly in legumes. In other words, individual plant foods such as cereal alone or pulses alone tend to be relatively deficient in one or more essential amino acids, and thus exclusive consumption of single plant foods such as chiefly rice or wheat would result in deficiency of an essential amino acid, and if this consumption pattern is continued over a long period of time, it can result in protein deficiency. However, a combination of plant foods, such as cereal-plus vegetable-based diets, are fully capable of meeting protein needs when consumed in amounts that satisfy energy needs. Fortunately, for us, the amino acid deficient in cereals, namely lysine, is present in ample quantities in pulses and green leafy vegetables. Similarly, the essential amino acid methionine, which is relatively low in pulse, is present in larger quantities in cereals. Thus, the different sources of vegetable proteins complement each other in terms of the amino acids they provide. Therefore, if we ensure that diet at every meal is a combination of cereals, pulses, and vegetables with nuts and milk contributing wherever one can afford, it will take care of the protein requirements. In this context, when we consider the Indian cuisine, we notice North and West Indian meals consist of chapati or rotis and rice as staples, eaten with a wide variety of side dishes like dals, curries, yogurt, chutney and achars. South Indian dishes are mostly rice-based, sambar, rasam and vegetables being important side dishes. The pulses legumes, included in the diet, with their high content of lysine but low content of methionine, complement the grains, cereals, which are more than adequate in methionine and cysteine but limiting in lysine. Such cereal pulse combination diets, when consumed, help the body receive all the indispensable amino acids. Hence, there is no need to worry about protein if we are eating a varied vegetarian diet. It is easy to get protein from lentils, dal, beans, curd, rice, soy milk, and cereals when eaten in combination so that their amino acid patterns become complementary. The inclusion of pulses in cereal millet-based diets is critical not only in increasing the protein content, but also in improving the nutritional quality of the protein. So, then, that brings us to the second question: can we improve the nutritive value of protein? Yes, as discussed above, the nutritive value of a protein can be improved in two ways. 1. By mutual supplementation, that is, by blending two or more proteins so that the excess of essential amino acids present in one protein makes up the deficiencies of the same amino acids in another protein, and 2. By supplementation of the dietary proteins with limiting essential amino acids. Let us understand these methods by way of few examples. 1. Mutual supplementation. Improvement of cereal proteins. Cereals, in general, are limiting in lysine and threonine, while legumes, milk, meat and fish are good sources of the amino acids. Hence, the proteins of legumes, milk, meat and fish supplement effectively cereal proteins. B. Soybean and sesame proteins. Soybean proteins are good sources of lysine but are deficient in methionine, while sesame proteins are good sources of methionine but are deficient in lysine. Hence, the proteins of sesame supplement effectively those of soybean. C. Improvement of cereal diets with legume and milk proteins. The proteins of poor diets based on different cereals are limiting in lysine and threonine, and their quality can be improved effectively by incorporation of legumes, soybean or milk proteins in the diet. So it is clear that mutual supplementation is the answer to improve the nutritive value of proteins. Another way to do so would be to supplement with individual amino acids as discussed next. 2. Supplementation with individual amino acids. Improvement of cereal diets by supplementation with lysine and threonine. Cereal diets supplemented with lysine alone or a mixture of lysine and threonine markedly increases the per of cereal proteins or proteins of poor cereal diets. B. Improvement of soybean and cow's milk protein with methionine. The proteins of soybean and cow's milk are deficient in methionine. Supplementation with methionine increases the per of the diet from 2.0 to 2.9 for soybean and 3.0 to 4.0 for milk proteins. C. Improvement of sesame and sunflower seed proteins with lysine. Supplementation with the limiting amino acid lysine increases the per of sesame proteins from 1.7 to 2.9 and sunflower seed proteins from 1.2 to 1.8. The discussion so far focused on methods to determine the protein quality and also on methods one could adopt to improve the quality of protein in a particular diet. Next, let us move on to protein requirements, how they are estimated and assessed. 4.5 Methods of estimating and assessing protein requirements at different stages of life cycle. In this section, We are going to deal with the methods that are used to estimate protein requirements, as well as the factors which affect it. Let's read and find out first what we mean by protein requirement and its significance. 100 years, human protein and amino acid requirements have been studied for well over 100 years using a variety of techniques. Nutrition scientists have collected data on the quantity of protein foods consumed in health, growth, and weight gain of various populations. The assumption was made that whatever healthy people ate was probably what kept them healthy and should, therefore, be used as a standard of comparison for other diets. These standards with respect to protein were invariably high for populations having an abundance of meat, milk, poultry, and fish in their diets. Wharton Atwater around the turn of the 20th century found intakes of 118 and 125 g protein day, respectively, for an adult woman and man. As nutrition developed as a science, more accurate methods for assessing nutrient needs were developed. Among these methods were those for assessing the intakes and excretion of nitrogen compounds. The Kjeldahl method, about which we learnt above, and other methods for determining the nitrogenous end products of metabolism were devised. These methods made possible the development of the concepts which today's scientists use to determine the nutrient requirements of humans, as well as other species, in protein nutrition. It was realized that the body consists of two pools of protein, one which has a short half-life and which must be constantly renewed, and one which is slowly broken down and rebuilt. If one assumes that over a short period of time, the pool having the long half-life contributes almost nothing to the nitrogenous metabolic end products, and then a measure of the amount of nitrogen excreted will reflect only the turnover of the short-lived proteins. These proteins have to be replaced by proteins newly synthesized from the amino acids provided by the diet. Hence, the term protein requirement means that amount of protein which must be consumed to provide the amino acids for the synthesis of those body proteins irreversibly categorized in the course of the body's metabolism. The intake of nitrogen from protein must be sufficient to balance that excreted. This basic concept is called nitrogen balance. This concept is useful in understanding the minimal need for protein in the diet. Protein requirement is greatly influenced by many factors such as age, environmental temperature, energy intake, gender, micronutrient intake, infection, activity, previous diet, trauma, pregnancy, and lactation. Let us take up each of these factors in detail. 1. Age. Protein in excess of maintenance needs is required when a new tissue is being formed. Certain age periods, when growth is rapid, require more dietary protein than other periods. Age differences in protein turnover, as well as protein synthesis, explain some of the effects of age on protein needs. Premature infants, those born before their 10 lunar month gestation time, growing at a very rapid rate, require between 2.5 to 5 g protein kg day if they are to survive. Studies of full-term infants have indicated that a protein intake of 2.0 to 2.5 g kg day resulted in a satisfactory weight gain, and that further increases in protein intake did not measurably improve growth. Older infants and children, whose growth rate is not as rapid as the premature or newborn infant, require considerably less protein, 1.25 g kg day. As growth rate increases during adolescence, the protein needs increase. Again, this can be related to the demands for dietary amino acids to support the growth process. As the human completes his growth, the need for protein decreases until it arrives at a level which is called the maintenance level. It is at this level that the concept of body protein replacement by dietary protein applies. During the growth period, it is very difficult to separate the requirements for maintenance from those of growth. The impulse for growth is so strong that it will occur in many instances at the expense of the maintenance of body tissues. For example, malnourished children will continue to grow taller even though their muscles, as well as other tissues, show evidence of wastage due to dietary protein deficiency. Growth carries with it not only a total nitrogen requirement but also a particular amino acid requirement. Maintenance, on the other hand, appears to have only a total protein requirement. The adult can make a number of short-term adjustments in his protein metabolism that can compensate for possible inequities of imbalances in amino acid intake as long as the total protein requirement is met. The young growing animal is not that flexible. The essential amino acid requirements are age-dependent. Although histidine can be synthesized in sufficient quantity by the adult to meet maintenance needs, yet it is not synthesized in great enough amounts to support growth or tissue repair. Thus, histidine is an essential amino acid for the infant, growing child, and injured adult. This is due to the nature of the growth and repair processes. Let us move on to the next factor and find out what environment influences have to do with our protein requirement. 2. Environmental temperature. As environmental temperatures rise or fall above or below the range of thermic neutrality, animals begin to increase their caloric expenditure to maintain their body temperature. In environments that are too warm, vasodilation, widening of blood vessels, occurs along with sweating and increased respiration. All of these mechanisms are designed to cool the body and all require an increase in the basal energy requirement expressed as per unit of body surface area. In cool environments, vasoconstriction and shivering occurs in an effort to warm the body and prevent undue heat loss. Again, an increase in basal energy requirement is observed. Smuts, 1934, found that nitrogen requirements were related to basal energy requirements. Through the study of a large number of species, he concluded that 2 milligrams nitrogen were required for every basal kilocalorie required when the energy requirement was expressed on a surface area basis. Thus, any increase in basal energy needs due to a change in environmental temperature will be because of the relationship between protein and energy. An increase in energy needs would be accompanied by an increase in the protein requirement for maintenance. In addition, profuse sweating as occurs in very warm environments carries with it a nitrogen loss which must be accounted for in the determination of minimal protein needs. The next factor that require attention in case of subjects selected for estimating protein needs is information regarding the previous diet, i.e., the kind of diet consumed by subjects in the past. 3. Previous diet. The effects of previous diet in the determination of protein requirements may be rather profound. If, for example, the subjects selected for studies on protein needs have been poorly nourished prior to the initiation of the study, their retention of the protein during the study will be greater than would be observed in subjects who have been well nourished prior to the initiation of the study. In other words, malnourished subjects have a higher protein requirement than well-nourished subjects. This of course raises the issue of whether there are body protein reserves. Watt, Wilson, Cuthbertson, Fisher, 1890, and others observed that animals fed on a protein-free diet exhibit a lag before their nitrogen excretion level is minimized. During this phase, the animal is metabolizing his protein reserve. Other investigators maintain that every protein in the body has a function, and if some of these proteins are lost, there is a loss in body function. Support for this concept is seen in the reduced ability of protein-depleted animals to fight infection or respond to the metabolic effects of trauma. Whether one believes that there is such an entity as a protein reserve may depend upon whether one perceives a difference between personal opinions on how nutrient requirements should be defined. Some nutritionists believe in stating the absolute minimum requirement to sustain life and then adding on increments for each body function above mere survival. This is known as the particulate approach. Other nutritionists believe that one cannot separate and quantify the individual requirements of each function beyond survival. They advocate a protein intake sufficient for optimal function of the animal. This is known as the integrative approach. The particulate and integrative approaches each have their merits when argued intellectually. However, since humans do not merely exist, Many human nutritionists tend to take the integrative approach to human nutrition requirements in their determination of protein needs. Next, we move on to another important factor that determines our protein needs, that is, physical activity. 4. Physical activity. Research on protein needs for muscular work had its beginning in 1863 when von Lieberg postulated that muscle protein was destroyed with each contraction of the muscle. On this basis, he recommended that heavy muscular work required a heavy protein diet. This theory has been amply disproved, yet even today many believe that a protein-rich diet will contribute to athletic prowess. Today, we know that muscle contraction does not result in destruction of the muscle. It, however, requires energy in the form of ATP, glucose and fatty acids, and does result in the breakdown of creatine phosphate to creatine, which is then converted to creatinine, a nitrogenous waste product excreted in the urine, as the energy requirement is increased to support the increase in muscular activity. So too is the protein requirement in much the same manner as described above for the effects of temperature. In a number of studies, the athletic performance of subjects could not be directly related to the quantity of protein consumed above that determined to be the requirements. Their muscular deficiencies were reduced unless a vigorous training program was included as a part of the experiment's protocol. Since most of the studies were of short duration and since muscle protein has a relatively long half-life, the lack of any demonstrable effect of protein intake on muscle performance is not surprising. Other factors such as sex, pregnancy, lactation, and trauma affecting the protein requirements have been studied. As can be anticipated, males, due to their greater physical activity and larger body size, have a larger protein requirement than females. Pregnancy, lactation, and trauma increase the protein requirements. So, now you realize that a large number of factors play a role in determination of protein requirements. Let us next find out what are the recommended allowances for proteins for different age groups. 4.6 Nutritional requirements and recommended allowances for proteins and amino acids. Human protein requirements are determined by methods such as A. N balance, and B. Obligatory N losses adjusted for efficiency of protein utilization. Amino acid requirement, indicator amino acid oxidation. IAO method is a method of measuring amino acid requirement. It is based on the concept that if one amino acid in the diet is below requirement, I limiting, then all other indispensable amino acids cannot be fully utilized for protein synthesis and the excess is therefore oxidized. With increasing intake of the limiting test, amino acid IAO will decrease, reflecting increasing incorporation into protein. Once the requirement for the limiting amino acid is met, there will be no further change in the indicator oxidation. It is a robust, rapid, reliable, and invasive method to determine amino acid requirement in neonates, children, and adults. The estimated average requirement of each essential amino acid is present in Table 4.5 earlier. Factorial method for arriving RDA for proteins of Indians, basal or obligatory N loss. The factorial method of assessing protein requirement of an individual can be in terms of obligatory loss of N through faeces, urine and skin. The obligatory loss of urinary and fecal N has been assessed by maintaining an individual on a protein-free diet and estimating the fecal and urinary N excretion. Total obligatory N loss for Indian adult subject, 48 ng/kgd. In terms of portion, this obligatory N loss corresponds to 48 ng x 6.25 is equal to 0.3 g portion kgd.
However, the efficiency of utilization of protein being relatively low at about 47%, the requirement of protein would then be the amount required to replace the obligatory loss divided by the efficiency of utilization. This would be 0.64 g protein kg day, which is similar to the median requirement of 0.66 g protein kg day. In addition, assuming ACV of 12.5%, the safe intake would be 0.83 g protein kgd. Factorial estimates of protein requirements for 0-18Y. The average protein requirement, APR, for years 0 to 18 is calculated as the sum of maintenance requirement plus protein deposited. APR is equal to maintenance deposition efficiency of utilization. Maintenance is calculated assuming that maintenance in young children is 0.58 g/kgd, which increase at 6 months to the adult value of 0.66 g/kgd. In the case of infants below the age of 6 months, the efficiency of utilization of protein for growth was assumed to be 66%, while beyond that age, it was assumed to be 58%. The protein requirement for growth of infants and children are given in Table 4.7 respectively. Table 4.7, Safe level of protein intake for infants aged less than 6 months. Table 4.8, Safe level of protein intake for children over the age of 6 months up to 10 years. Look at the screen. Protein requirement during pregnancy and lactation. Protein requirement during pregnancy has been assessed by the factorial method as the additional requirement for fetal growth and expansion of maternal tissue. Protein requirement during pregnancy is given in Table 4.9. For a gestational weight gain, GWG, of 10 kilograms, the protein deposition would be 2.5 gd and 5.3 gd for the second and third trimesters respectively. Table 4.9. For a GWG of 12 kilograms, if protein deposition was assumed to be proportional to GWG, a deposition rate of 3.0 gd and 6.4 gd is calculated for the second and third trimesters, respectively. Protein deposition rates of 2.0 gd and 4.3 gd for the second and third trimesters respectively were calculated for a GWG of 8 kilograms. The safe levels for additional protein intakes were thus calculated to be 7.6 gd and 17.6 gd respectively. Table 4.9, Recommendation for additional protein intake during pregnancy for 12 kilograms. 10 kilograms, gestational weight gain. During lactation, protein requirement has been computed on the basis of secretion of 6.4 g per day of protein in milk during 0 to 6 months and 5.0 g during 6 to 24 months. Protein requirement during lactation is given in Table 4.10, Lactation ERS. Table 4.10, Additional protein requirement during lactation, based on N balance. Nitrogen balance is the difference between nitrogen intake and the amount excreted in urine, feces, and sweat, together with minor losses occurring by other routes. To estimate the protein requirement, levels of dietary protein below and near predicted adequate intake are fed and nitrogen balance is measured at each level. The requirement is estimated by interpolating and extrapolating the nitrogen balance data to the zero balance point, nitrogen equilibrium, for adults or to defined level of positive balance to allow for growth for children. Several studies on healthy, adult Indian subjects have shown that the minimum dietary protein intake for an equilibrium ranges from 0.5 to 0.66 g protein kg day with a mean of 0.58 g kg day, which after allowing for integumental loss of 11 mg n kg day, works out to 0.65 g protein kg day. Assuming ACV of 12.5%, the safe value of intake, mean 1.96 xsd, works out to 0.81 g protein kg day. The corresponding requirement based on a mixed Indian diet will be 0.98 g protein kgd, and this figure is also close to a kgd. Note, for people consuming predominantly cereal-based diets with low-quality protein, the protein requirements are 1 g kgd. for men the protein requirement would be 65 gd and for women 55 gd. The cereal-legume milk composition of a diet for a moderately active man should be 3.1 to 0.5 to meet daily protein requirement. After having a thorough knowledge of the factorial method of estimating N requirements, surely you would be now in a position to calculate protein requirements. The nutritional requirements of protein for Indians in different age groups and physiological stages as suggested by the ICMR is given in Table 4.11. Table 4.11, Summary of Recommended Protein Requirements for Indians. Protein requirements can also be expressed as ratio of protein energy to total dietary energy requirement, expressed as protein energy ratio, pay ratio. Pay ratio, therefore, describes the protein-energy interrelationship. Protein utilization and deposition are dependent on intake of adequate energy. Adequate non-protein energy from carbohydrate and fat is essential for dietary amino acid to be utilized for protein synthesis and for amino acid-related functions in the body. If adequate dietary energy is not available, dietary protein is inefficiently utilized. Pay ratio will differ for different ages and also between individuals engaged in different activities, lifestyles. Refer to Table 4.12. It presents the pay ratio for different age groups. Safe recommended intake of protein is expressed as the ratio of recommended energy intake, Table 4.12, protein energy ratio, percent, for different age groups. 4.7 Protein deficiency. One of the most common nutritional disorders in the world today is the deficiency of protein. Both adults and children are affected, as the populations in the less developed nations of the world exceed their food supply. Due to the ubiquitous nature of protein and its role in bodily function, protein deficiency is characterized by a number of symptoms. In many situations, not only is protein lacking in the diet but also calories. For this reason, it is difficult to segregate symptoms due solely to protein deficiency from those of energy deficit. In children, one may observe the different symptoms and visualize them all as parts of a continuum called protein energy or protein calorie malnutrition, PEM or PCM, rather than distinctly different nutritional disorders. Kwashiorkor was the term used to describe a disease first observed in the Gold Coast of Africa by Dr. Cicely Williams in 1935 and at first was regarded as a dietary state where only protein was deficient, not energy. Marasmus, on the other hand, was regarded as a dietary state where both protein and energy are deficient. Now it has become apparent that the symptoms of any one of the twin diseases may intermingle with the other so that a clear-cut diagnosis is impossible. You look up the unit now as you go about reading regarding PEM here in this unit. Here a very brief overview of the disorder has been presented. A. Kwashiorkor. The term Kwashiorkor means the disease the first child gets when the second baby is born. That is, the sickness of the deposed child. Thus, the disease could be cured by milk. Kwashiorkor usually affects the young child after he is weaned. The child is usually between 1 and 3 years old and is weaned because the mother has given birth to another child or is pregnant and cannot support both children. If the child has no teeth, he is given the gruel. This may be a fruit, vegetable or cereal product mixed with water and hence not a good protein source. Can you think of a few reasons which could limit or decrease the protein intake? Let us see what these are. Cultural food practices or taboos may further limit the kinds and amount of protein given to the child. Apart from this, concurrent infections, parasites, seasonal food shortages and poor distribution of food amongst the family members may also contribute to the development of Kwashiorkor. The deficiency develops not only because of inadequate intake but also because at this age the growth demands for protein and energy are high. The symptoms of kwashiorkor are as follows: growth failure. This is manifested by decreased body length and low body weight in spite of retention of water in the body, edema, and presence of subcutaneous fat in some children. This growth retardation is primarily due to the general quantitative lack of proteins. Mental changes. Several workers have stressed on the constant finding of mental changes described as apathy and peevishness. In advanced cases, children tend to live in an inert, listless condition and show no interest in the surroundings. Edema occurs at first in the feet and lower legs and then may involve the hands, the thighs and face. The edema is mainly due to lowered serum albumin and probably also due to high sodium and low potassium levels in serum. There is also some evidence that the normal diuretic-antidiuretic hormonal control of urine secretion gets disturbed. Muscle wasting. Muscle wasting is a constant feature of kwashiorkor, and a reduction in the circumference of the upper arm is usually evident. It is less affected by edema than in the forearm or leg. Moon face. The full, well-rounded face, known as moo face, is often present in kwashiorkor. Liver changes. Liver is slightly enlarged and fatty infiltration of liver is usually present. Gastrointestinal tract, loss of appetite and vomiting are common. Diarrhea is present in most cases. Skin and hair changes. The characteristic skin changes of kwashiorkor are known as the crazy pavement dermatosis. This is most marked on the buttocks, back of thighs and axil. These lesions consist of dark hyperpigmental brownish-black areas of skin. Anemia. Anemia is invariably present. It is due to the deficiency of iron and folic acid. Anemia may be aggravated by parasitic infection, which prevents the absorption of nutrients. Vitamin deficiency. Signs and symptoms of vitamin A deficiency such as xerophthalmia and keratomalacia are widely prevalent. Angular stomatitis and glossitis due to deficiency of riboflavin may be present. Biochemical changes. Several biochemical changes have been reported in children suffering from kwashiorkor. Serum albumin. The serum albumin content is usually low, ranging from 0.7 to 2.2 g/100 ml. Serum albumin level is a good index of the severity of the disease, and the rise in serum albumin level during treatment is a reliable index of the rate of recovery. Enzymes in serum and digestive juices. Low levels of choline esterase, alkaline phosphatase, amylase, and lipase have been reported in kwashiorkor. In experimental animals fed on protein-deficient diet, reduction in lipase, amylase and protease activities of pancreas have been reported. Let us learn about marasmus next. B. Marasmus. Although children of all ages and adults can suffer from deficiencies of both energy and protein, the marasmic child is usually less than 1 year old. In developing countries, a common cause for marasmus is the cessation of breastfeeding. Milk production by the mother may have stopped because of her poor health, death or deliberate decision of the mother to bottle feed her baby. This decision might have a socio-economic connotation. Can you think of it? Well, it is just that the mother may view bottle feeding as a status symbol, or she may be forced to work to earn a living and may be unable to have her baby with her. Or she may not be able to lactate. While under optimal conditions of economics and sanitation, it seems that the bottle-fed child may be well fed in emerging nations, but this is not always true. The mother may not be able to buy the milk formula in sufficient quantities to adequately nourish the child. She may over-dilute the milk, or she may use unsafe water and unsanitary conditions to prepare the formula for the child. This plus the insufficient nutrient content often precipitously leads to the development of marasmus, a form of starvation characterized by growth failure with prominent ribs, a characteristic monkey-like face and matchstick limbs with little muscle or adipose tissue development. Tissue wastage but no oedema is present. Whereas the kwashiorkor child has a poor appetite, the marasmus child is eager to eat. The child is mentally alert but not irritable. Anemia and diarrhea are present for the same reasons as in kwashiorkor. The skin and hair appear to be of normal color. Protein hormones, which regulate and coordinate the use of dietary nutrients, are not found in adequate amounts. Cell hormone receptor sites are affected, which further dampens the effectiveness of the hormones produced. Marasmic and kwashiorkor children have decreased blood sugar levels, decreased serum insulin and growth hormone levels, and in marasmus, decreased thyroid hormone levels. The clinical features of marasmus are as follows. Nutritional marasmus. Nutritional marasmus is principally due to the consumption of diets markedly deficient in both proteins and calories. It is seen most commonly in the weaned infants of about 1 year of age in contrast to kwashiorkor, which occurs more often among children of the age group 2 to 4 years. Nutritional marasmus usually is precipitated by diarrheal diseases. Clinical features. The two constant features of nutritional marasmus are growth retardation and severe wasting of muscle and subcutaneous fat. Growth retardation. This is usually very severe. Loss of weight is much more marked than decrease in height. The child is usually below 60% of the standard weight. Wasting of muscle and subcutaneous fat. The subject is severely emaciated. The muscles are wasted. The arms are thin and the skin is loose. Subcutaneous fat is practically absent. Other changes. The skin is dry and atrophic. The subject shows signs of dehydration. Eye lesions due to vitamin A deficiency and anemia may be present. Biochemical changes. There is a slight lowering of serum albumin. Vitamin A content of serum is low. The important difference in the clinical and biochemical features between marasmus and kwashiorkor are given in Table 4.13. Table 4.13. Clinical and biochemical features of marasmus and kwashiorkor. What is the treatment of kwashiorkor and marasmus? The treatment of both kwashiorkor and marasmic children requires care and caution, as their enzymes for digestion and their protein absorption and transport systems are less active. Feeding these children with large quantities of good quality protein would be harmful. Well, this might sound strange to you, but it is so. Their diets must gradually be enriched with these proteins to allow their body sufficient time to develop the metabolic pathways to handle a better diet. Giving these children solutions of either predigested proteins or solutions of amino acids may be of benefit initially, but these solutions too must be used with care. If the amino acids in excess of immediate use are deaminated, and if the pathway for synthesizing urea is not fully functional, ammonia can accumulate in the child and become lethal. We have studied about kwashiorkor and marasmus as conditions typical of protein energy malnutrition. But, interestingly, a large number of cases show signs and symptoms of both marasmus and kwashiorkor. These are the intermediate forms. Let us learn about them next. Marasmic kwashiorkor. In countries where the incidence of protein-calorie malnutrition (PCM) is high, a large number of cases show signs and symptoms of marasmus and kwashiorkor. These intermediate forms are called marasmic kwashiorkor. In addition, the interrelationship between the two major syndromes is such that the changing circumstances may result in a transition from one clinical picture to another. A child with early kwashiorkor can develop nutritional marasmus by severe infective diarrhea and ill-advised prolonged underfeeding. Conversely, an infant with nutritional marasmus may develop kwashiorkor if fed on protein-deficient carbohydrate-rich foods along with adequate common salt. With this we end our discussion on the consequences of protein deficiency, as well as other varied aspects of protein which may influence human health. In the next unit we shall discuss about lipids in detail. However, you must attempt the check your exercise below before reading further. 4.8 Summary. This unit covered the important macronutrient proteins. Proteins, you learnt, are vital to all body cells, tissues, organs and functions of organ systems. The process of digestion, absorption and metabolism of protein are complex and involve several nutritional and non-nutritional factors. Proteins are widely distributed in nature, the richest vegetarian source being soybeans. Egg protein has the highest biologic value. Several methods can be employed to determine the protein content of foods, the most popular being those based on growth and body weight changes. Protein quality in the diet may be improved by mutual supplementation of protein-rich foods and cereals. Nutritional requirements of proteins vary with age and physiological activity stage. Deficiency of proteins leads to the twin disorders of marasmus and kwashiorkor and may be cured by dietary intervention. Thank you. Subscribe to our channel for more updates, and we will see you with the next chapter.